चल ठीक। Radioactive bacteriophages are allowed to attach to E. coli bacteria. Then infection viral coats were removed from bacteria by agitating them in a blender. How they were removed? By agitating them in a blender. Were now separated by spinning them in a centrifuge. Virus separated by spinning them in a centrifuge. Bacteria which was infected with viruses that had radioactive DNA were radioactive. Bacteria which was infected with viruses that had radioactive DNA were radioactive, indicating that DNA was the material that passed from viruses to bacteria. Bacteria were infected with viruses that had radioactive protein but were not radioactive. This indicates protein did not enter the bacteria from viruses. DNA, therefore, the genetic material is passed from bacteria to virus. This paragraph states that bacteria which was infected with viruses had radioactive DNA were radioactive মানে जो बैक्टीरियाটা was bacteria infected with virus रे radioactive DNA थिला radioactive DNA therefore कौन शो करला that DNA पास ହେଲା ଯେହେତु ପ୍ରୋଟିନ୍ ନଥିଲା radioactive ସେୟା कि एटा देने बैक्टीरिया रेडियोएक्टिव एस लेवल प्रोटीन कैप्सूल रेडियोएक्टिव डीएनए तो बैक्टीरिया रे रेडियोएक्टिव डीएनए मिलला नो रेडियोएक्टिविटी इन सुपरनेटल चला और नो रेडियोएक्टिव एस रेडियोएक्टिविटी इन सुपरनेटल ठीक प्रॉपर्टीज ऑफ जेनेटिक मटेरियल डीएनए वर्सेस वायरस नॉट सो इम्पोर्टेंट बट ओके लेट्स कंटिन्यू From the following discussion, it is clear that the debate between proteins versus DNA as genetic material was unequivocally resolved by Hutchinson's experiment. It became established that it is the DNA that acts as a genetic material. However, subsequently it became clear in some viruses. RNA is the example of tobacco mosaic virus, QB bacteriophage. Answer to some of these questions: why DNA is predominant whereas RNA? So why DNA is predominant? Because molecule that এটা important board question molecule why which are the criteria for molecule to act as DNA. It should be able to generate its replica. First of all se nijo replica generate karibo. Stable should be stable chemically and structurally stable thiba achi. Provide scope for slow changes. Mutation heba achi not that evolution kon slow process thae setpa semti yaad rakho je slow mutation hebo. Express itself in form of Mendelian character. Eta best eta important statement based question. If one examines each requirement one by one, because of rule of base pairing and complementarity, both our nucleic acid, DNA and RNA have the ability to direct their duplication. The other molecules in the living system such as protein fail to fulfill this criteria first. The genetic material should be stable enough not to change with different stages of life cycle like age or with stability is one of the properties which was very evident in Griffith. Stability was one of the property which was evident in Griffith itself that which killed the bacteria. At least did not destroy some of the properties of the genetic material. ठीक कौन लगा जिस ग्रिफिथ रो एक्सपेरिमेंट थिला ग्रिफिथ रो एक्सपेरिमेंट ए एक्सपेरिमेंट रे जेनेटिक मटेरियल टा चेंज होला लाइक तारा जेनेटिक कैरेक्टरिस्टिक टा चेंज होला stability of as one of the properties of genetic material was evident if Griffith transforming principle itself that heat which killed the bacteria at least did not destroy some of the properties of genetic. Heat which killed the bacteria did not destroy. This can easily the light of the DNA, the two strands separated by heating. This can be explained that in the light of DNA, the two strands being complementary if separated by heating come together when appropriate conditions are provided. Further, two is present at every nucleotide in RNA. Further, 2' OH group is present at every nucleotide, which is a reactive group which becomes RNA liable and easily degradable. Therefore, RNA is also now known to be catalytic, hence reactive. RNA is now known to be catalytic assertion. Why it known to be catalytic? Because 2H group present at every nucleotide in RNA, a reactive group that makes RNA liable and easily degradable. Hence reactive. Therefore, DNA is chemically less reactive and structurally more stable. In fact, the presence of thymine at place also confer additional stability to DNA. Both DNA and RNA are able to mutate. RNA being unstable mutate at faster rate. Therefore, RNA genome and having shorter life. Viruses having RNA genome have shorter lifespan. RNA can directly code for synthesis, hence can now easily express it. DNA is dependent on RNA for synthesis of protein. Protein synthesis machinery has evolved around RNA. RNA world important.